So finally, our third speaker is Dr. Paul Burridge. He's an assistant professor in the Department of Pharmacology at Northwestern University Feinberg School of Medicine, and he's the founding faculty member of the Center for Pharmacogenomics. He completed his PhD at the University of Nottingham and postdoctoral fellowships at Johns Hopkins University and Stanford University. Dr. Burridge has worked on with human pluripotent stem cells for nearly 2 decades, with a primary focus on the application of these cells with disease modeling, and specifically the pharmacogenomic and molecular mechanisms of chemotherapy-induced cardiovascular toxicity. So Paul, you're going to speak on the role of the genome in doxorubicin-induced toxicity. We look forward to your lecture.
Okay, great. Thanks for that introduction. Sorry, it's a bit of a mouthful. So here is a continuation. So some more calcium imaging of iPS cell-derived cardiomyocytes. So I'm going to talk exclusively about our in vitro model using iPS cells to model doxorubicin cardiotoxicity. So you've already had an introduction to this. From a in vitro model standpoint, the important thing to know about doxorubicin is if you give a patient a high enough dose, then they essentially will certainly have cardiotoxicity. In vitro, that's really important because that means we can just give the cells a very high dose of doxorubicin, and then we know what our cardiotoxicity phenotype will be. Of course, in patients, mostly we're talking today about pediatric patients. With solid tumor patients, commonly get 450 milligrams per meter squared. So when the patients all get the same dose, the real issue for us is, why do some patients have toxicity and some don't? And as you've already seen today, there's a strong indication that that's genetic. So moving on to our model, we published 5 years ago now this paper in Nature Medicine in which we show if you take some patients who have had doxorubicin and then had cardiotoxicity, and then you make some iPS cells from them and turn them into cardiomyocytes, those cardiomyocytes, indicated by the red stick figure here, those cardiomyocytes are more sensitive to doxorubicin in vitro. So here is our kill curve, and as you can see, for the iPS cell cardiomyocytes derived from the 4 patients who had toxicity, their cells die at a much lower dose of doxorubicin. And intriguingly, for patients who didn't have toxicity, they actually seem to be slightly resistant to doxorubicin. Essentially, those cells can survive a slightly higher dose when compared to healthy patients. So that's patients in this case who didn't have doxorubicin at all, and we don't really know what their phenotype is. So on the right here is actually data from the first time we ever did this experiment, showing that these are iPS cell cardiomyocytes from the 4 patients, and as you can see, with increasing doses of doxorubicin, the cells start to die. And so this was the first time we'd ever shown that these stem cell derived cardiomyocytes accurately recapitulate the patient's individual predisposition to toxicity. So in many ways, this is also the first demonstration that this predisposition is actually genetic, because the iPS cells, the only thing they get from the original patient is the genome. Everything else, they lose their epigenetic state, and really it's just the DNA that is transferred. So that raises a number of very interesting questions. So if the iPS cell cardiomyocytes are more sensitive to the doxorubicin, the question is, what aspect of the cardiotoxicity are these cells recapitulating? Now, doxorubicin cardiotoxicity has 4 major mechanisms that occur. One is that the doxorubicin comes into the cells. It essentially hijacks the mitochondrial enzymes and then uses those to produce reactive oxygen species So we showed that for patients who had toxicity, they had higher levels of reactive oxygen species. There's also DNA damage involved. We showed for patients who had toxicity, they have DNA damage at a lower dose of doxorubicin. There is, as you just saw from Javier's talk, there is calcium overload, so essentially mishandling of calcium that then leads to damage to the sarcomeres, so sarcomeric disarray, which you can see here. So again, patients had toxicity, their iPS cell cardiomyocytes have that at a lower dose. And then finally, there's mitochondrial dysfunction. So in doxorubicin cardiotoxicity, mitochondrial dysfunction is kind of a part of all of the different mechanisms. If you have DNA damage, then that's usually sensed by a mitochondria-related process. Obviously, the reactive oxygen species is related to mitochondrial function as well. And just like all of the other mechanisms, we show that the iPS cell cardiomyocytes recapitulate that too. So this was a little disappointing for us. We assumed that the iPS cell cardiomyocytes would recapitulate one specific function of the cardiotoxicity pathway, but actually it seems that they model all of the different parts of the mechanism. So that's it's really useful, but also it makes it much more difficult for us to quickly get to the answer of, what is the genetic differences between these patients who had toxicity and didn't that are causing this doxorubicin sensitivity. So you've heard a little bit today. This is a figure from a review we did a while back. So you heard about the CBR genes and SELF4. This is all the genes that have ever been associated with doxorubicin cardiotoxicity, ranked by p-value, essentially how likely we think they are. So both SELF4 and CBR are high on this list. But for us, we were interested in the most associated variant. So that's one in RARG. And then the second variant in SLC28A3, which is still very highly associated, but importantly, the only variant that's ever been replicated. And that by that, I mean, it's come up in 2 different studies. Everything else usually only comes up in 1 study. So we started off looking at this gene RARG. Sorry, so like I was saying, SLC28A3 is the only gene that's been replicated. SLC28A3 is a transporter. So we naturally hypothesized that SLC28A3 might be able to transport doxorubicin into cardiomyocytes, and therefore might be, your expression of this particular gene between 2 different populations might be responsible for the level of doxorubicin that gets into cardiomyocytes. So for this project, what we did was we went to our collaborators in Vancouver and got them to re-recruit some patients from their original study so 3 patients who do not have the SNP of interest, and then 3 patients who do have the SNP of interest. And then after an absolutely vast amount of work, you can see this comparatively subtle difference in their response to doxorubicin. So patients with the variant are actually protected from doxorubicin. And that seems to make sense if you think this is an inward transporter and the variant makes that transporter less efficient. So if you have the SNP of interest, your SLC28A3 is not as effective, and therefore it can't bring as much doxorubicin into cardiomyocytes. And that's also, we see less programmed cell death too. So we knocked out and overexpressed this SLC28A3 using a CRISPR-based system. And in a similar manner, you can see if you knock out SLC28A3, the cells are protected from the doxorubicin cardiotoxicity, and if you overexpress this gene, they are more sensitive. And that makes you think that, again, they might be taking up more drug. Conveniently, doxorubicin is autofluorescent. That means with a flow cytometer, you can actually detect the amount of doxorubicin that gets into cells. So in our iPS cell cardiomyocyte model, we can see with the overexpression, they do have more doxorubicin, and with the knockout, they do have less doxorubicin. So that was really interesting, except in this particular scenario, and quite commonly from genome-wide association studies, the SNP of interest doesn't change the amino acid sequence, which makes things difficult. What that actually means is the SNP that has been chosen to be represented on these chips that they use to do SNP analysis actually probably is not the effector variant. It is merely co-inherited with the effector variant. So that means it's a tag SNP. So just a variant that is commonly occurs along with a number of other variants. So we did this analysis where we essentially looked at our SNP of interest, other SNPs that are co-inherited, which is called in linkage disequilibrium, and then also of those SNPs, so these are all the SNPs that are in linkage disequilibrium, and then of those SNPs, which ones are in areas that are involved with chromatin regulation, and then also which ones are in regulatory regions. And this turned out to be really interesting because our SNP of interest doesn't come up on this, but this new one, which we're going to call RS111 for now, seems to fit all of these three different categories. So we went from having a piece of information where we really didn't know whether the SNP did anything at all, to finding that its relative, its co-inherited variant that actually is in a really interesting area. So I shan't expect you to be able to read these very easily, but this is the SLC gene. It's in reverse orientation in the genome, so it's expressed in this direction. And right at the end of the gene, again, in the opposite orientation to the SLC28A3, is what is now called SLC28A3 associated sequence. So we have had this name registered. This is a long non-coding RNA, and our SNP that we're interested in, RS111, is right in the middle of the first exon of this long non-coding RNA. So these are all the other SNPs, and as you can see, they're kind of in introns or not in very interesting regions, and comparatively this one seems to be in a really interesting region. So we did some work overexpressing and correcting this particular SNP of interest. So this is using an even more advanced CRISPR technology, where we essentially take lines that have this 111 variant and we correct them versus the ones that innately have it. And again, what you can see is if you correct this variant, the cells, essentially the SLC28A3 then becomes much more functional again, and the cells start taking up more doxorubicin. So we can also express this long non-coding RNA individually, which does reduce the level of expression of SLC28A3. And now you're used to these LD plots, you can see that the expression of this long non-coding RNA is pretty substantially, significantly, I mean, protective from doxorubicin cardiotoxicity, and reduces the uptake of doxorubicin by around about half. So that was really interesting to us. So we now know that there's a long non-coding RNA that controls the expression of SLC28A3, and we think that's one of the major players in the uptake of doxorubicin. Transporters are usually very druggable. Because they transport things, it also means it's comparatively easy to get drugs into them. So we've done this screen of all FDA and EMA approved drugs, and we discovered these 10 drugs that seem to be protective from Doxorubicin Cardiotoxicity. So this is a completely unbiased screen. And one of the drugs that came out is this drug, Doxorubicin. And we found that Doxorubicin seems to be very protective from cardiotoxicity. So Doxorubicin is an old-fashioned tricyclic antidepressant. Through a huge amount of in vitro modeling, in silico modeling, we know that Doxorubicin does bind SLC28A3. It's essentially a competitive inhibitor, and that treating the cells with Doxorubicin before they have Doxorubicin essentially is cardioprotective in a similar manner to a knockout of SLC28A3. So to cut a long story short, this is in a mouse model. If you pre-treat a mouse with Doxorubicin before it has Doxorubicin, you attenuate the cardiotoxicity by about half. We have a lot of data on this. We're pretty convinced that that's a real result. And then the cool thing about SLC28A3 is it's never expressed in cancer cells. It's very specific to the heart. It is expressed in other tissues, but it's definitely not expressed in cancer cells. So we've screened a huge number of cancer cell lines, including a lot of sarcoma lines, and show that if you pre-treat them with Doxorubicin, it doesn't attenuate the toxicity of Doxorubicin whatsoever. So the short answer is, we now think that Doxorubicin cardiotoxicity can be predicted genetically. As you see, it's polygenic. There are a lot of variants associated with it. And I think many of these variants we've discovered in the past are going to lead us to potential drug mechanisms. So our recommendation is that patients should be screened for variants in SLC28A3, especially this RS111 that we are interested in. In this case, what do you do if the patient has a variant? Right now, clinically, I think if patients were screened for that, it would be the kind of standard cautious approach to Doxorubicin therapy, such as enhanced echo and other methodologies. But yes, we are right on the point now where we're really interested in Doxorubicin. It is an FDA-approved drug. One would argue that giving that to patients who are undergoing chemotherapy would be comparatively trivial. Obviously, we need to do more work on confirming that it doesn't have any negative effect on the chemotherapy efficacy. But all of our data to date suggests that's pretty good. So I'd just like to thank the people who did the work. Most of this work was done by my postdoc, Tarek. And I'm happy to take any questions. Thanks.
Good. Well, thank you, Paul, for a wonderful lecture.